Hello, everyone. Dr. Welsh here from Lindenwood University in Missouri, United States of America. Today I am recording a little lecture. This is an example lecture of what we would like to see from each of the groups, where you discuss a monster from ancient folklore and a modern day disease, which could be an equivalent of. Today I'm going to be using my examples, a ghoul and the disease known as Kuru. Now, what is a ghoul? A ghoul comes from old ancient stories. Depending upon where you're at in the world, it could go back quite far. To define what a ghoul is, in modern day parlance, we term it as the undead who are digging up and eating the bodies of the dead. Or, depending upon which comic book, which movie you're watching, they're the undead who are making dead people that they will then turn around and eat. Now, when we look at the folklore, we see that this goes back quite a long time. We see mentions of things that were ghoul or ghoul-like in ancient tales, ancient Arabic tales and Persian tales. So this goes back quite a ways. This isn't a modern day invention. As we see right here, you see right there, if you look in ancient Arabic stories, going back, 1,000 years more, we'll see that instead of ghoul, as we refer to it now, it's gul and gula. Gul if it's a male undead, gula if it is a female undead. Modern interpretations we see within the past few decades, especially in the mid-late 20th century, Lovecraft brought these back. H.P. Lovecraft and his Cthulhu and his ancient gods. We see it real big now in comic books, as well as in movies and video games. The walking undead, not the zombies. Zombies are different. These are ghouls. These are going to be things that are inhabited by spirits and whatnot. So when we look at ghouls and ghoul-like, these entities, as I said, have been around in Arabic stories for over millennia, going back BC, AD time and forward. What we would think of as ghouls, they saw more as, this is their early interpretation of jinn. Jinn is what we think of here in Western Europe and the Western societies as the genies. The jinn weren't these three magical wishes granting things. These were more demons. They would attack and eat humans. And over the years, as different populations moved around, different cultures kind of merged and things, what was the jinn became more ghoul, as Arab societies, Persians who also had these ideas of these, moved into Eastern and then Western Europe. It became more and more of what we would think of the stories as ghouls. You can see here, sorry, I keep pointing the wrong way. You can see here, this is an illustration from an illuminated manuscript. I am going to mess up this pronunciation. For those of you who have come from these countries, Persian countries, I apologize. The Shamana. This was written and illustrated. This version was handwritten and illustrated, illuminated, back around 977 to 1000 AD. You can see here the king, Rostam, is fighting a ghoul as part of one of his 7 activities, one of his 7 victories. Here in Western European societies, we refer to this shamana as the legends of kings. So you can see it goes back quite a ways. Here we also see from the story of the Arabian Nights Entertainment, the collection of stories we think of as the 1001 Arabian Nights. It's actually a short story collection of different stories. This one happens to be the picture you see here. Sorry, did it again. Picture you happen to see here is a wood print from the book The Arabian Nights Entertainment was published in 1840s in London. This is actually Anim discovered with the ghoul. Anim was an evil witch. It was cannibalistic witch, I should say. And you see the man there that she's looking at is actually a ghoul. They are feasting on that dead body. And up to the top there, you can see Sidi Numan. Sidi Numan is actually, the history of Sidi Numan is the name of the story. Modern day interpretations, like I said, early 20th century with H.P. Lovecraft, early to mid, we see H.P. Lovecraft with his Cthulhu stories and whatnot, brought a new interpretation to this. Instead of the undead, these are more hominids now, humanoids from alternate realities or from alternate universes who have escaped into ours. And you can see from the picture here, this is an interpretation of one of H.P. Lovecraft's ghouls. It almost looks human-ish, but it feasts on us, feasts on the dead. And as I said, starting mid 20th century on to present day, we see more and more ghouls and ghoul-like things, the interpretation showing up in movies, comics, and video games. Now, if we switch up a bit and we look at what could be diseases that we would misinterpret, that our forefathers from long ago would have misinterpreted and just said, oh, those ghouls or ghoul-like things. Well, 4 of them shown here could be African sleeping sickness, rabies, what we now refer to as Hansen's disease, which was always previously referred to as leprosy, and then Kuru. Kuru is the one I'm going to be focusing on. Okay, Kuru, it's more correctly referred to as transmissible spongiform encephalopathy. Its name tells you exactly what's happening. It's an encephalopathy, meaning infection of the brain tissue itself. You have infection and inflammation of the central nervous system. Spongiform tells you what it does. The damage it does is it causes little small pockets of death, decay, cell death within the brain. So when the early scientists were going through and researching why certain people died with certain symptoms, signs and symptoms, during the post-mortem, the autopsy, when they removed the brain had these pockets of death, little necrotic, little bubbles. And they just said it looked like a sponge, hence the name spongiform. Transmissible because it is transmissible human to human. Okay, so this could be direct human to human, human, animal, back to human, depending upon which form you're talking about. We're going to see, for those of you who have background in prions, Kuru is closely related to bovine spongiform encephalopathy, what we are commonly referred to as mad cow disease. And it's also closely related to Creutzfeldt-Jakob disease, CJD, which is another transmissible spongiform encephalopathy. Now, what we see, though, the difference between Kuru and Creutzfeldt-Jakob disease is, we're going to see over the next few slides, Kuru starts with physical signs, symptoms, ailments, and then progresses to dementia. Whereas Creutzfeldt-Jakob disease, the dementia hits much earlier on in the slow chronic progression of the physical signs and symptoms. So there is a bit of a difference going on there. Either way, when you look at it, it's a disease of the central nervous system. Okay, you're going to see physical as well as neurological effects that are going to slowly, chronically build over time. Time here could be weeks, months, years. Kuru was first reported in 1957 in the Foray tribe found on one of the islands of Papua New Guinea. When they, so for the past few decades, as we've been studying this, we see that the signs and symptoms of the disease presentation slowly accumulates. So we see different stages of the disease progression. The first is this ambulant You'll see ataxia, cerebral ataxia. People have trouble, like I apparently am today, and talking, recognizing words, recognizing people, recognizing things, and making this connection. What is what I see, and this is the word describing what I see. They start to lose that. They start to lose the central to peripheral nerve system interaction, so their gait's going to change. They're going to be shivering as they muscle control seems to be slowly degrading. And the cerebral implications, the neural pathways are not firing correctly, or they're going to be misfiring. So you're going to start to see hallucinations. You're going to see inability, like I just said, see this, know the word for it. That's going to start to break down. The second stage is going to be a sedentary stage. The inability to stand unsupported. So they're now losing peripheral nervous system control. Neuromuscular junctions are now not working correctly. So the brain is trying to send signals, but signals either aren't leaving the brain. Wow, trying to talk fast and it's not working. Trying to leave the brain, and either not jumping from central to peripheral nervous system, or they're not making it down the peripheral nervous system to the neuromuscular junction. So the ataxia, that's getting worse. Remember, ataxia, being able to see and know word, and point out what you see. Oh, this word means this thing that I'm going to point to. That's lost there. Loss of fine motor control, fingers, eyes. Their eyes start twitching in different ways because they can't control which way they're going to be looking. And then finally, the third stage is also known as the terminal stage. Here, bedridden because they have complete paralysis, flaccid paralysis. They can't move, they can't control. There may be some flailing, but it's uncontrollable. Almost to the verge of very advanced MS. You've seen that with people with MS, muscular, so that's the kind of things you're going to see there as it goes from the sedentary to the terminal. Dysphagia, inability to swallow. So you're going to see a lot of weight loss as they go from sedentary to terminal because they lose the ability to swallow. They can maybe chew, they can salivate, but they can't get the food from the mouth to the stomach. So they're going to start going through, and they're going to be losing a lot of weight. Let's just go with that way. And then incontinence. They're going to be losing sphincter control, not only at the anus but also in bladder. Diagnosis for this, beyond just this chronic progression of the disease, you're going to have to wait till postmortem. I'm going to have to look at the autopsy and examine the brain tissue histologically to look for the spongiform. Now, the causative agent of what we call Kuru, this is going to be a prion. Okay, we're going to talk about this. We're going to talk about these three things: prion, the metabolic interactions of what the prion do, and its transmission, and is followed by treatment. So the prions, as you can see here, prions on the left, every neuron, every nerve cell in your body and many other cells in your body make a protein known as PRP superscript C. Okay, this is the gene product of the gene PRNP, as you see there, capitalized and italicized, because remember that's how we denote proteins versus gene, whether it's all capital, whether it's italicized or not. And then to the right there, B, that's what happens to the PRPC protein. It gets misfolded. So instead of being a bunch of alpha helices at the tertiary structure level of the protein, it now becomes more beta sheet-like. The misfolding of the protein will then lead to what you see here. Instead of being individual PRPC, they now become PRPSC. They become flattened, beta strand-like, and then they start accumulating, aggregating. And it's those aggregates in the cytoplasm, the aggregating at the plasma membrane, that leads to the issues we see with Kuru, leading to the spongiform encephalopathy and whatnot. Now, when you look here, you'll see that PRPC has quite a few roles in the neurons. PRPC, the native protein, the normal, quote unquote, normal native protein, actually is a plasma membrane protein. It is made in the cell, transported to the plasma membrane of the neurons, and it has a role. It interacts with numerous transport proteins. It helps regulate multiple transport proteins. It's a little hard to see here, but two of them that I think are of utmost importance. A calcium channel. Remember, our cells, calcium in the cytoplasm is not always good, but you want to have some, but not a lot. So you're going to be pumping calcium out extracellular, or you're going to be pumping calcium into the endoplasmic reticulum. So that way you keep very low levels of any in the cytoplasm. So PRPC, native protein, will interact with the calcium channel to help regulate when it opens, when it closes. It's also going to interact with a glutamate receptor. Okay, you want to be able to pump in, pump out glutamate. It's going to be one of these things that it's moving across into and out of the cell is going to trigger different cellular responses. And you can look over there to the far side and you'll see that there are numerous other receptors, and not only the receptor, but the downstream signaling cascades that are dependent upon the activity of PRPC. Now what happens, under normal conditions, PRPC, the native protein, is synthesized in the ER. It's embedded in the ER membrane. It's going to be trafficked to the Golgi using a transport vesicle. In the Golgi, you're going to get the final twist, tweaks, cuts, pastes that always occur in the Golgi. Final. And then it's going to be transported to the plasma membrane. And it's going to sit there in plasma membrane, and it's going to interact with the receptors I just talked about on the last slide. Now, on the flip side of that, PRPSC, the prion, is taken up in a vesicle, or I'm sorry, it's not taken up. It's taken up in a vesicle because an extracellular PRPC protein is going to interact with one of the native proteins embedded in the plasma membrane. It's going to force a refolding of PRPC to PRPSC, the prion form. This will then trigger uptake via clathrin-coated pit. It'll go around, it'll be taken up in the cell, and then distributed in vesicles where it's then going to interact with other PRPC native proteins, and refolding them into the PRPSC prion form. And then all of these will either be exported out to re-release exocytosis, or will now be on the surface of the plasma membrane of that neuron, where, as you see here, they are going to interfere with different receptor causing signal cascades. The signal cascade, PRPC-SC, is going to interact, and it's going to interfere with one of the proteins, the deactivation and thus the activation of BIP. BIP is one of our proteins that's there. It's held in check unless the cells become damaged or in danger, and then BIP starts the cascade that will lead to apoptosis. Okay. PRPSC there also interacting with BIP can also interfere with ER chaperone protein. So it's going to mess with the transporting from ER to Golgi, Golgi to the ER and all of that, which also signals a signaling cascade, which will ultimately lead to apoptosis. So there's multiple ways that apoptosis, because PRPSC is going to be in the plasma membrane, interfering with, as it shows here on the right, calcium channels, or glutamate uptake channels, or others, and they're all going to result in apoptosis of that neuronal cell. Now, it's not just that one cell, because there's going to be a release of PRPSC proteins that are made inside. Those vesicles are going to be released, exocytosis, and then it's going to spread to neighboring neurons or neighboring Schwann cells, and it's going to spread and spread and spread. So that's within the person transmission. And what we find is PRPSC is not just neuronal. We find, brain, other neurons, other nerve cells throughout the body can also be infected by PRPSC, leading to more spread throughout the body. It's just the accumulation of the effects is more visible, signs and symptom-wise, if it hits the central nervous system, specifically the brain. Treatments, there's really not that many treatments right now. There are some that are in development. The problem is you really don't know you have Kuru or Creutzfeldt-Jakob disease or whatnot, or some of the others known prion diseases until you start present neurological damage. Once you start presenting neurological damage, the signs and symptoms we associate with neurological damage, specifically with Kuru or Creutzfeldt-Jakob disease, the damage is already done and there's no reversing. What they are looking at, as we're showing here, there are different compounds that either in vitro, in a test tube, or in a petri dish on neurons can show that they can inhibit, if not reverse, the PRP to PRP-SC, so they can revert it back or they can stop it from happening. And they're now progressing into mouse models. If we inject PRP-SC into the brain of a mouse, can we use any of these different compounds or a cocktail of these compounds to inhibit the spread? So yes, you're going to lose some neurons, but the neighboring neurons are not going to be affected. And that's the state of it right now.